Hello everyone. Today we will learn a special complex in immunology. The name is MHC. Their complete name is Major Histocompatibility Complex. So let's take 2 questions to learn this content. Also, we take these 2 questions in the following chapters. So first, why we have the problem for graft rejection? How about the beginning of immune response? Equal to how the foreign things, actually antigen, make the immune response happen. So let's take these 2 questions, and in the following sections we learn. So let's see how the scientists found the MHC complex. So at the beginning, the scientists did experiments. So let's see this experiment how they did. So now we have 2 strains mouse. So in simple, one is white mouse, the other is black mouse. So now we want to transfer the skin from the black mouse to the white mouse. So here, the mouse B to mouse A. So after 14 days later, and we found the skin become necrosis. So what happened? Immune response happened. So now continue, we transfer the black mouse to this white mouse, and after 6 days later, around 6 days later, and we found the skin become necrosis. So the time is shorter. That means this mouse has the immune memory. So now we have this naive A mouse. So now we inject the lymphocytes to this mouse. That means we transfer the immune memory to this white mouse. And also, we give this mouse the skin from mouse B. So around 6 days later, we found this mouse cannot accept this skin, and this skin become necrosis. So now we know. So immune response mediated this reaction. So the scientists want to know which kind of antigen make the mouse have this problem. So these 3 great scientists, and they found this complex. And later, in 1980, they got the Nobel Prize. So the red scientist Snail, he used the mouse to find which kind of antigen or which kind of molecule decide the transplantation rejection. So he named this gene is histocompatibility genes, and he found the second histocompatibility genes is major responsible for the transplantation rejection, and he gave the name is H2 in mouse. And in the middle, the scientist and Dosette, he found one kind of molecules expressed on the leukocytes, and he gave the name is human leukocyte antigen. So also decided the transplantation rejection. And this left scientist, he found some MHC genes in this molecule. So let's think in 1980s, the Nobel Prize gave this prize to the scientist who did the great research for the MHC. So let's see the concept of MHC. What is MHC? So let's see. So every mammalian species studied to date possesses a tightly linked cluster of genes. So here, a tightly linked cluster of genes. So we gave the name is the major histocompatibility complex, and this complex has the function as to for the T cell recognition and to discriminate the self and the non-self. So now, first we know this concept. Later I will explain. And also we can name the MHC or give the concept of MHC is a cluster of genes encoding polymorphic cell surface molecules. So this surface molecules is a major histocompatibility antigen. So for mouse, it's H2, and for human, it's HLA, and also involved in with the T cell and in transplantation rejection. So this is the concept for MHC. From concept, we know. So we have the antigen, and the genes is in a very tightly linked cluster of genes. And their function is for the recognition by T cell and also for the transplantation rejection. Because just now I introduced the experiment from the mouse, and we know because of this antigen exists, we have the problem for the transplantation rejection. So now first, today, this topic we learned some genes of the MHC. So both for mouse and for human, we divide these MHC genes to 3 groups. So class 1, class 2, and class 3. So for mouse, class 1, we have K loci and D or L loci. And for class 2 for mouse, we have the I loci. This is for human. So for human, for class 1, we have B, C, A, 3 loci, and for the class 2, we have DP, DQ, DR, these 3 loci. And this gives us the genetic map of MHC genes. So let's think about its concept. So these MHC genes located in a very narrow region in the chromosome. So for mouse, it's chromosome 17, and for human, it's chromosome 6. And let's see some characters for the MHC. It's very interesting. So for the MHC, the genes, they were co-dominant expression. So we know some genes is dominant and some gene is recessive, right? So although you have this gene, but it is recessive, maybe we cannot see its appearance. But for the codominant expression, that means we have the genes from our parents, so from mother, from father, but we can express all the genes on our cells. So this for the mouse, and this is for the human. So now let's think, if for human, for HLA, we have A, B, C, 3 loci, and we have 1 pair from our parents. So that means at most for the human body, we have 6 class 1 molecules expressed on our body. That means at most we have 6 class 1 molecules expressed on our cells. And here we can see the class 2, because later we know they have 2 chains. So these genes can cross to each other. So for human body, for the class 2 molecules, at most we have 12 class 2 molecules expressed on our body. So now from the codominant expression, so use the inbred mouse, we can have this, we can find this phenomenon. So this the inbred mouse, and this is from parent, and this their first generation. So this first generation express all the genes from their parents. So what will happen if we do the skin transplantation? So if the first generation, that means can accept all the skin from their parents. But if we give the progeny, this skin to their parents, no, the parents cannot accept the skin from their kid, from this progeny. So this is because of the codominant expression for MHC molecule. And this figure shows us, so from the generation, from the parents, and we can give the MHC molecules to their progeny. So for their progeny, so they have only half same as their parents. So if we want to look for the donors for transplantation, it's better look for from their brothers and sisters. And sometimes they can have the recombination for their MHC from their parents. But normally, the HLA gene will transfer as a haplotype, that means as a unit to transfer to next generation. So now that think what we learn about the genes for the MHC. So this is the concept. So each mammary species. So for rabbit, we call them RLA, and for the pig, we call it SLA, and for human, special name is HLA, and for the mouse is H2. But all these genes actually is MHC. So what is MHC? That means all the mammary species to date possess this a tightly linked cluster of genes. So these genes located in the very narrow regions in the chromosome, and they will have the function is the intercellular recognition and also discrimination between self and non-self. So we will learn this part in the following sections. And let's see for the MHC molecules. And first, the MHC is composed of multiple genes. So that means in the population, the MHC is the most diversity. And each set of alleles is transferred to as a haplotype. So normally we get 2 haplotypes from our parents. And an individual inherits 1 haplotype from the mother and 1 haplotype from the father. And the last one is very interesting, is that the alleles is codominant express. So okay, this we learn for the first part.